Don't let the sun catch you lying at my front door.
Oh, hey there. I was just getting oxidized. It's Mr. Reardon. It is now Tuesday, November the 4th, and I want to take another crack at our conversation about the light-dependent reactions and what's happening on the thylakoids. After working with First Block this morning, I realized I made a couple of mistakes on my mind map, and I'm kind of seeing some clearer ways to explain things. So I want to take another crack at a talk that I recorded yesterday that you'll probably never see because I want to upload it to YouTube. But the big idea today is how it is that thylakoids can harness light energy and convert that into ATP. And as always, our screencasts are brought to you by a beverage. Today it's water. Water, the source of electrons for the light-dependent reactions. Water. Well, as I said in class, I'm not crazy about these diagrams that we have in Campbell. Now, they're beautiful. I'm sure the artist is super talented, but I find that when you try and look at these photosystems together in the thylakoid membrane, you can't really tell what's going on from an energetic standpoint. So I wanted to take a minute and look more closely at how photosystem 2 and photosystem 1 work in tandem, and really just wanted to focus on flow of electrons, how water is used, and then discuss briefly the concept of chemiosmotic production of ATP. And again, a lot of that is just review from last year. And again, this isn't supposed to be a standalone lesson. This is really, the idea here is to bridge what we know from, bridge what we talked about in class and give you a second crack at it. So here, what I want you to see in this mind map that I developed is stripping away all the phospholipid bilayers, stripping away all the individual proteins that make up a photosystem, and really just trying to concentrate on the essentials. So what I want you to imagine right here in this green box is the reaction center of photosystem 2. This is as we call, a special chlorophyll A molecule because it sits inside that photosystem. And then above it, there is a protein, which here it's purple. We call it the primary electron acceptor. That is photosystem 2. To its right, we have another photosystem, again, reaction center, a chlorophyll A molecule, special because of its placement. This one is excited by red light here at a wavelength of 700, hence we call it P700. And again, another primary electron acceptor. So the idea here in class, like if we just oxidize chlorophyll and allow those electrons to be boosted by a photon, when they fall, you guys, they're going to release a photon of light. And what's super cool is that light will be red, just like we saw in class in our little demo today when we took kale, put it in acetone, allowed those membranes to denature, and then shined a light through the pure chlorophyll supernatant. And what we got was a red light. But what happens in photosynthesis, you guys, is as that chlorophyll A molecule in the reaction center is excited, or as it's oxidized, the electron is boosted, and then that electron is held. Once that electron is held here by the primary electron acceptor, it has potential energy, and we can do something with that energy. And that's what I want to talk about right now. So what I want you to see before we look at the raw materials that enter photosystem 2 is get a feel for these yellow lines. These yellow lines represent the flow of electrons. So electrons move from photosystem 2 to a primary electron acceptor through this purple block. That is an electron transport chain, you guys. And then those electrons tumble down towards photosystem 1, where they are excited again by another wavelength of red light, 700 nanometers, and those electrons are boosted to another primary electron acceptor. And then they can be used. And so what you're looking at here is 1, 2, 3 paths that one little electron takes. We call that non-cyclic flow of electrons because it begins at one place and ends at another. When I was your age, we call this Z-line photosynthesis because the pathway looks like a Z, or like an N. I don't know why they didn't call it inline photosynthesis, but what do I know? Well, I know a lot, but I don't know why they don't call it inline photosynthesis. Then the other thing I do want you to notice, so we've got this non-cyclic flow of electrons, and then you'll also notice that some of the electrons, they may move off over here to the right. We'll explore that in just a minute. But other electrons will tumble back through an electron transport chain back to photosystem 1, where they get boosted again, and fall back, and boosted again, and fall back. Well, if the previous flow of electrons was non-cyclic flow, you can guess what this flow of electrons is called. You guessed it, cyclic flow of electrons. Pretty clever. All right, so we got electron flow. We'll talk in just a second about what that electron flow is used for, but before we do that, let's take a closer look at photosystem 1. Photosystem 1 is a busy, busy place. I want you to think back to August. I want you to think transpiration. I want you to think bulk transport of water from the soil to the roots, up the xylem, into the mesophyll, and now that water is entering the chloroplast of a functioning cell. It is here where water is truly used by the plant. When a photon of light at 680 nanometers strikes the reaction center of photosystem 2, that chlorophyll A molecule is oxidized. It loses an electron. That electron is replaced, you guys, by an electron from water. So here at photosystem 2, water enters the picture. The solar energy splits water into pieces. The electrons are boosted to the primary electron acceptor. The hydronium ions are released into the stroma, where they're going to be pumped into the thylakoid space in just a second, and that oxygen is also released into the stroma. Well, oxygen, atomic oxygen, is very unstable, and it likes to partner up with another atom of oxygen. So as water is split, atomic oxygen forms molecular oxygen, and O2, what we know and love, let's all breathe it in, 21%. That oxygen is given off as a byproduct. So where does the oxygen come from in photosynthesis? It comes from the splitting of water, and all of that happens at photosystem 2. Let's look a little further. That electron gets boosted, it gets held, but just like anything that gets held, it wants to fall. The beauty of the light-dependent reactions is that those electrons don't just fall down. What happens is they actually tumble through, and I will show that to you in just a second. They're going to tumble through electron transport chains, and those electron transport chains will use the energy generated by the falling of electrons to pump hydronium ions against their concentration gradient, create a chemoelectrical gradient or an electrochemical gradient, and then tap that electrochemical gradient to make ATP. This is chemiosmosis. So think about everything you know about mitochondria. Go watch my little talk on electron transport chains and oxidative phosphorylation inside the mitochondria, and imagine that that same thing is happening along the thylakoid membrane of the chloroplast. And again, these electrons tumble down electron transport chains. That energy is coupled to the chemiosmotic production of ATP. But wait, there's more. When those electrons rest at photosystem 1, another photon of red light, here at wavelength 700, so a little bit longer wavelength, a little bit lower energy, is going to boost that electron. Those electrons will some electrons will tumble back through these electron transport chains, generating a little more ATP, while others are associated with yet another electron transport chain and an enzyme called NADP reductase. Now, NADP reductase, it's a very creative name. Can you guess what that enzyme does? You guys are so smart today. NADP reductase reduces NADP to NADPH. It is reduced. It is gained electrons. And these, I talk about it in class, but these are the hydronium ions that are used to reduce carbon dioxide to sugar, G3P. So in here I say that right here. The protons and the electrons associated with NADPH, these are the protons and electrons that are used to reduce carbon dioxide to glyceraldehyde 3-phosphate in the Calvin cycle. That's the material. Well, where does the energy come from? The energy that came from the electron transport chains was coupled to the chemiosmotic production of ATP, and that ATP is used to drive these non-spontaneous reactions in the Calvin cycle. The energy from ATP, I'll pull it over, the energy from ATP is used to take the protons and electrons from NADPH and reduce carbon dioxide into a functioning sugar. The fate of that sugar and what that sugar is used for will be a discussion of another podcast or screencast. Thanks so much for watching. I'm going to get back to being oxidized, and I will see you guys in class.
Don't let the sun catch you lying at my front door.